Today, we're diving into the incredible journey of a biologic drug. It's a decade-long, billion-dollar marathon that takes a single idea born in the lab and transforms it into one of the most advanced medicines on the planet. I mean, have you ever really thought about it? This isn't like baking a cake with a simple recipe. It's a mind-boggling process that blends biology, engineering, and just an unbelievable amount of precision. So let's pull back the curtain and see how it's done. So first things first, what is a biologic drug? You know, when you think of a normal pill, you're thinking about chemistry, mixing chemicals together. Well, this is completely different. A biologic is a super complex molecule, like a protein, that's actually made inside of living cells. It's really less like chemistry and more like farming on a microscopic scale. So where does this microscopic farming operation begin? Well, it all starts in a research lab, with scientists trying to create the perfect genetic blueprint for a cure. This is really where the magic happens. First, scientists identify the bad guy in the body that's causing a disease. Then, they design a therapeutic molecule that can fight it. The next step is to take the gene that makes this molecule and insert it into a host cell. A lot of the time, they use something called a CHO cell. That's a Chinese hamster ovary cell. These things are the absolute workhorses of biotech. From there, they screen thousands of these new cells to find the superstar, the one that's the most stable and pumps out the most protein. That superstar cell is then used to create a master cell bank. Think of it like the original gold master copy of a record. Every single dose of this drug, forever, will come from this one pristine source. Okay, so we've got our master blueprint. Now what? Well, it's time to fire up the factory. And I mean that literally. We're heading inside the living factory. You can really think about this whole factory process in two big steps. First, there's upstream. That's where you grow trillions of cells and get them to make the protein you want. And then there's downstream, which is basically the massive high-tech cleanup job of purifying that one protein from everything else. So for the upstream part, picture these huge, shiny stainless steel tanks called bioreactors. Inside, billions, even trillions of our engineered cells are growing. It's like a 5-star hotel for cells in there. Everything is perfectly controlled, the temperature, the pH, and a special nutrient broth that's like a gourmet meal. The goal was simple: keep those cells as happy as possible so they churn out huge amounts of our target protein. Now, after the cells have done their job, we're left with this complicated soup of stuff. The downstream process is all about fishing our one specific protein out of that soup. So how's it done? Well, first, they filter out all the cell factories themselves. Then comes the really cool part: chromatography. It works like a molecular magnet. A special material inside a column grabs only our protein, letting all the other junk wash away. Finally, they concentrate the good stuff and run it through one more filtration step just to be absolutely sure it's pure and safe. So, we've made the pure drug. Awesome. But now comes the hardest, most expensive, and riskiest part of the entire journey: proving it's safe and effective in humans. We're talking about the billion-dollar gauntlet. Before any drug can get near a patient, it has to survive years of these grueling clinical trials. It happens in stages. Phase 1 is really small, with just a handful of healthy volunteers. And it's all about one question: is it safe? If it passes, Phase 2 expands to a few hundred patients to ask, okay, does it actually work? Then comes Phase 3, the big one. We're talking thousands of patients. This is the final exam, to prove on a huge scale that it's both safe and effective. And even after all that, Phase 4 keeps an eye on the drug for years once it's out in the real world. And the cost to successfully navigate this gauntlet, it's just staggering. Bringing a single one of these drugs to market can easily cost hundreds of millions of dollars, and sometimes it tops $1 billion. It is by far the biggest hurdle in this whole process. All right, let's say our drug is a superstar. It made it through the gauntlet. Well, it's not over yet. Now it has to face the final bosses, the regulators, who give the final green light. Companies have to package every single piece of data they've collected over the past decade into a massive submission. It's called a Biologics License Application, or BLA. Regulatory agencies like the FDA then go through this mountain of information with a fine-tooth comb. They check everything: the data, the manufacturing plants, you name it. Every single claim has to be backed up by cold, hard evidence. And once they get that green light, production kicks into hyperdrive. We're now talking about those commercial bioreactors that can be over 10,000 liters. That's bigger than a cement mixer truck. And the quality control at this stage is just off the charts. Every single batch has to be a perfect identical clone of the drug that was tested in those clinical trials. We are so close. The drug is approved, it's being made by the truckload, but how does it get from that giant factory into a vial that a patient can actually use? This is the critical fill-finish stage. The pure drug gets mixed with special stabilizers to make sure it lasts. Then, inside an incredibly sterile environment, we're talking air cleaner than you can possibly imagine, automated systems precisely fill each vial or syringe before it's sealed up and ready to go. And here's something you might not think about. These biologic drugs are fragile, really fragile. Most of them have to be kept in a super strict temperature range, usually between 2 and 8 degrees Celsius, from the moment they leave the factory to the moment they're given to a patient. This whole unbroken, refrigerated supply line is known as the cold chain. And if it breaks at any point, the drug is ruined. It's that serious. You know, when you step back and look at the whole thing, from that first cell to the final vial, this quote just nails it. It's not just one scientific discovery. It's this amazing fusion of deep biology, massive-scale industrial engineering, and incredibly strict regulation, all working together over a decade or more. This whole journey, this is the absolute cutting edge of medicine as we know it today. And it leaves you with a really fascinating question, right? If this is what we can accomplish now, what's next? What will the medicines of tomorrow even be capable of?